Hi, good morning, good afternoon. Welcome to the EMBODY Proposers Day. We're going to talk through a number of things today, and primarily we're going to talk about the EMBODY program. And so with this, I can turn it over to our director, Amy Jenkins, who will talk about ARPA-H goals and the mission of ARPA-H. So, Amy.
Hi, everybody. Good morning, good afternoon. I'm just here for a few minutes today, as you can see on the agenda, to welcome everybody, to give you a little bit of an overview of ARPA-H, our goals, and to also talk a little bit about the office that Daria and this whole program resides in within ARPA-H. What I really want to do is welcome this potential community of performers to ARPA-H. This is obviously a new organization, fairly new organization within the Department of Health and Human Services here within the US government. And all of you here today are now kind of some of the people that we'll interact with in the future through this program and through many others. So welcome, and excited to tell you about EMBODY today. So let's see, look at this. Everything's moving smoothly here today on our webinars. So again, thank you all for joining us, and I'm excited to tell you a little bit about the ARPA-H model. So as many of you may know, ARPA-H was stood up in the spring of 2022 by President Biden. We had our inaugural director announced in the fall of 2022, October 2022. So we've been really running for about 18 months now. And the organization was stood up with the business practices of DARPA in mind. So some of you may be familiar with DARPA. And DARPA is the Defense Advanced Research Projects Agency, and was stood up in 1958 in response to the launch of the Sputnik satellite. And so this created a lot of concern across the country about how could there be this crazy thing that nobody understands up in the sky. And DARPA was stood up with a new business model that aimed to prevent and create strategic surprise in the national security space. So what can we do from a national security perspective to make sure that we're never surprised again, but that we are always creating the surprise? Out of that agency, many innovations have come, like the internet, self-driving cars, stealth technology, and then more recently, the real accelerator on mRNA vaccines. What we're trying to do here at ARPA-H is to apply that same model to health. So what can we do in the health space, in the healthcare space, to really create healthcare of the future, things that are going to surprise us in the future? That's what we want to try to facilitate here. One additional thing that I'll point out is that oftentimes the efforts that we invest in at ARPA-H, we may not always know what the end result is going to be. I will say that I can assure you that the program manager that was first involved in that ARPANET did not envision the internet of today. They did not set out to create what we have today, but they did set out to make sure that these computers could talk to each other. And so some of the things we do here today and in the coming years are only yet to be imagined by hopefully our grandchildren in the future. So we have a very simple mission statement here at ARPA-H. It is to accelerate better health outcomes for everyone. We take that mission statement very seriously. Many of the things that we'll do in this office are going to be things that may not be realized for many decades, but we are always thinking about everything we do with the eye towards how is that going to accelerate health outcomes. As I mentioned, President Biden stood up the office or the agency in March of 2022. And a lot of people ask the question, what is the organizational structure of ARPA-H? So ARPA-H is an agency within the NIH. We are not an institute or center within the NIH. The ARPA-H director reports directly to the HHS secretary, not to the NIH director. But we are administratively linked to the NIH, and that has actually really been very, very advantageous for us. We have a lot of contacts within the NIH, and it really allowed us to hit the ground running as an agency 18 months ago. We have no internal research labs. We are a funding agency only. We are also disease agnostic, so we're not the National Cancer Institute. We will fund efforts in cancer, we will fund efforts in specific diseases, but we have no mandate, and our funding is not tied to any one particular disease. As a funding agency, it's important to have appropriations, and we do have an initial $2.5 billion initial appropriation from the government, and our appropriation is independent of the NIH budget. Another thing that many of you who have read the draft ISO are now very intimately aware of is that we do not fund grants. So we fund typically contracts, other transactions, or cooperative agreements, but we don't fund grants. We are not a grant funding agency. And we do have some unique ability to interact with the FDA. So what I want to talk about, we have four specific mission offices across ARPA-H. These four offices are outlined here, and three of them are grayed out, not because they're any less important, but because we're all here today for the Health Science Futures Office to hear about the EMBODY program that lives within the Health Science Futures Office. So I'll start from the other side of the slide and just work very quickly back. We have three other offices: Resilient Systems, Proactive Health, and Scalable Solutions. You can very quickly see here the types of initiatives that are going to be funded through those offices. Scalable Solutions, how can we reach everybody quickly? In Proactive Health, keeping people from becoming patients. And Resilient Systems, building a resilient healthcare infrastructure of the future. Many of you may say, well, it seems like I could, some of these things could live in Scalable Solutions or Proactive Health. The lines between these offices may oftentimes be blurry because this is healthcare, this is biomedical science, and we recognize that some things may fit in several offices, but there will be major thrust areas within each office that allows for our focus. And so what I do want to talk about a little bit here is the Health Science Futures Office. This office can arguably be labeled or talked about as the most broad office at ARPA-H, where we have a very simple remit, and that is to expand what is technically possible. So we're looking to develop the tools, the technologies, and the platforms of the future. We will have efforts that span from very imaging-based and physics-based approaches all the way through to translational efforts in surgery. And this allows this office to invest in some things that are, for lack of a more technical term, the really crazy out there ideas that we may not recognize their impacts for several decades. And I think many of you, as you have read the ISO, and as you're going to hear from Daria today, will recognize some of those concepts in the EMBODY program. Many of the things that Daria is asking you to do are pretty crazy. They're going to be hard, and that's what we're aiming to do within this office. I do want to address the healthcare ecosystem that ARPA-H functions within. So we have many federal agencies that we interact with as stakeholders, of course, the NIH, but many federal partners such as FDA, CMS, that we interact with on a daily basis. But the most important part of this slide is over on the left-hand side, and that's you all, the performers. These are the teams. We don't call you all grantees. We call the people that we issue awards to performers because we don't issue grants. But these are the people that we don't have internal labs. We want to do wild, crazy ideas. We really want to push science, but we don't do it. You all will do it through your great ideas. And so we often get the question of who can we fund at ARPA-H? Who are the groups that can be funded? And the answer is really all of the above. Large academic labs, large industry, small businesses, small labs. All of these are appropriate agencies and appropriate groups that we will fund within ARPA-H funding. Of course, we also want to take into account our customers. So we work very closely with patient advocacy groups, healthcare providers, the public. There may be some folks on the call today that fall into some of these categories. Generally, we restrict these proposers days. We're looking for the people that will be the doers on the program, but we don't want to ever exclude patient groups, healthcare providers, and the public from understanding what we're doing. And we have many other opportunities to interact with those groups. So I'm just going to very briefly talk about the Health Science Futures Office, give you a little bit more of a flavor of what we're doing within this office. Outside of the EMBODY program, you're going to hear a lot about the EMBODY program from Daria coming up in the next presentation. So as I mentioned, the Health Science Futures Office is investing in innovative technologies, tools, and platforms to enable healthcare of the future. This may look like novel systems to modulate the host. It may look like novel delivery systems with spatial and temporal precision. We want to ensure that anything that we do mitigates off-target effects in the wild and crazy ideas that we're going to come up with in this office. We often think about the types of things that we do in this office and how we get to those solutions by these general concepts here. So we want very adaptable approaches. We want people to be able to zoom out, think about what the big picture is, what we want to achieve on the end, and in order to make that possible, what needs to be true, what has to happen along the way scientifically. And then we build in those metrics into our proposals and into our programs. We want very innovative technologies. We want people thinking about things that you may say, wow, if I were to go to a conference of my colleagues and say, this is what I'm going to do, maybe 60% of the room may chuckle a little bit and say, that's not possible. That is okay. That is solidly the space that we want our performers and our programs to live in. We want people to question dogma. We want people to push the foundations of science. It may not work. It may fail. But if we don't push, we're not gonna hit those goals. And so we really want to put people outside of their comfort zone, and we're asking you all to be a little bit outside your comfort zone when you think about presenting these solutions to these programs. We really want to expand the possibilities of what the current solutions are. So again, not getting down and bogged down in dogma and saying, well, it didn't work 10 years ago, there's no way it's going to work now. There may be a new insight, a new enabling technology that allows us to believe that if we try this time, we may be successful. And another thing that you will hear a lot about today, and likely see a lot in the ISO, is that the things that we ask teams to do are very, very ambitious, and very rarely could any of the things that we put in these programs be achieved by one group. What we're looking for is many groups to come together into somewhat consortiums under one prime contractor that can propose a solution. But it's very rare that one team can solve all of the problems. And that is really why we are here today. We want you all to meet each other. We want you to hear from the program manager to know exactly what she wants, and she is going to be able to tell you exactly what she wants today. But we also want you to meet each other. We want you to talk to each other. We want you to say, hey, I've got a great approach to TA1, but I don't know what I'm doing with TA2, or I'm lost in this realm. I need somebody that can work on TA2, and come together, meet each other. That is the most important part of today, is finding that team that can come together to solve these big problems. I want to give a little bit of a flavor of what we're doing within the Health Science Futures office, both to welcome you all, see some of the other things we're doing in this office. We have launched 7 programs across our office since standing this up just over a year ago is when I started. So very excited to be able to have these 7 programs on a slide all together. So our very first program on Nitro is an osteoarthritis program. We've launched a surgery program in Psi, a computational approach to vaccines in Apex, eye transplantation in Thea. Many of you are very familiar with what's going to happen in EMBODY, low-cost, adaptable platform for in vivo production of cell therapies. I'm really excited about the potential of the EMBODY program, and very excited to be here today. And then our 2 newest programs, Print and Light, those have upcoming proposers days to both print organs in the print program, and then finally to develop the diagnostics for the lymphatic system in the Light program. So with this, really excited to have this portfolio. Excited to have you all here today to think about the problems that Daria is putting forward with the EMBODY program, and to think about how you can find your partners, people of the future that are going to help solve this big problem. I am going to very briefly talk a little bit more about our model here, just so that you all understand the urgency that you're going to hear out of Daria today. So our program managers are not permanent government employees. Our program managers come into ARPA-H on a term of 3 years with the opportunity to re-up their position for up to another 3 years, for a maximum of 6 years. That is the case for Daria, that is the case for all of the program managers in the agency, and that is the case for myself as well. So this agency will change every 3 to 6 years. Program managers will come in, they'll start programs, maybe 2 to 5 programs in their time here, and they will leave and they will hand those projects off to those projects and programs off to other program managers. But the program managers that are here, they have a very defined mission. They have a problem that they want to solve. Daria is putting forward the EMBODY program to solve a problem that she is very passionate about, but she will not be able to be here and watch this for in perpetuity. Hence, her putting very strict timelines, very strict metrics on this program to try to really hit the accelerator on these technologies and make this a reality in 6 years. This is particularly a nuance to our model. It makes us quite different than many other funding agencies in that our program managers have extreme urgency. They are going to push that urgency on you, and that urgency is what really accelerates fields forward in our experience at DARPA, and having translated that model over here to ARPA-H. Overall, the programs are stood up in this method. So our program managers come in, they identify a difficult health-related challenge, a very defined problem that they believe is ripe for solving given where the scientific field is today. They define that challenge. They don't want this to be something that is incremental or easily solved through things that exist today, but we're also not doing science fiction here. There needs to be things that are rooted in science, maybe some foundational efforts that have been published out of publications and efforts that have occurred at other organizations like the NIH and the NSF. That leads us to believe that if we put very defined, discrete funding around this effort, we'll be able to hit the accelerator on it and push it forward by decades. The program manager comes in, they pitch that idea to myself and the agency director, and then they get approved for a budget for that program. And so that program now launches, and that's where we are today. We are launching this program, telling the community of people that may work on this problem, here's the problem we want to solve, and here's how we believe we need to solve it. And then you all in the community will submit abstracts and proposals to your methods for your approach to solving this problem, and they will be reviewed by a panel by the program manager and by other government reviewers. And the program manager makes the final decision and says, these are the groups that I want to move forward with on funding of this effort. And these are the approaches that we think are going to be the most likely to succeed, knowing that there is always risk and there's no guarantee of success given the risky nature of what we're trying to do in these programs. These teams will work towards the goals of this effort for 6 years or 5 or 6 years, depending on the program. Sometimes the efforts may not work, and sometimes teams may have to stop. Their efforts may stop. There may be specific parts of a program that say, at this point, you will be reevaluated for next steps in the effort. That is program specific, and you're going to hear about Daria's approach to that when she talks about embodying specifically. But what we also want people to know is that we recognize that this is R&D, that this is going to be hard, that there are going to be need to change the approach sometimes. We don't want people to just say, okay, I have to take this approach because this is the approach I proposed. If a problem comes up, rework, rethink about a new path and try to take that other path. What we care about at ARPA-H is that end goal. We want that end exclamation point, that end capability that we're going to deliver to the healthcare sector. How we get there, the approach we take to get there, that's less important. What's most important is that we deliver that capability. So with that, I want to welcome you all once again. Thank you for being here today. I think you're going to hear some really exciting things about the EMBODY program. You're going to hear from several folks across the agency that are going to explain the inner workings on a technical side and also on a very programmatic contracting side as well. So thank you for all being here. And I am going to turn this over now to Dr. Daria Fedukina, who is going to tell you all about the EMBODY program and hopefully change the future with us. Thank you, everybody.
Good morning. So EMBODY, it stands for Engineering of Immune Cells Inside the Body. Okay, so first what I would like to do is to remind you where you can find information on the program and to talk about links that are useful. So first, we have ARPA-H website. This is a courtesy website where you can find useful information. If you go to this site, arpah.gov, and go to research and funding, And then in the program section, you will find the EMBODY webpage. And most of you have gone there because you're registered for the Proposers Day. But just to remind you that there is a link there for solicitation. And then there is also a link to ask a question. And you can continue ask questions. We will close this ask question option after April 22nd, but you can still ask questions even after this Proposers Day in case while you're reading the ISO and have something that you would like to clarify. If you click on this link that says EMBODY Special Notice and Draft Innovative Solutions Opening, it will take you to sam.gov. sam.gov is the primary source of information. All the updates are posted here, and you can see two links here as well. The first one, which says Draft ARPA-H SOL 2403. This is the ISO document that I would encourage you to read in detail. It describes everything you need to know about the program, and it's a downloadable PDF file. And I will give you today an explanation of the program and some screenshots from this document to sort of for ease of visual memory. Okay, so first what I would like to do is to talk about EMBODY vision, because it's very useful to know what it is as you're preparing solution summaries and think about your proposals. EMBODY program aims to make engineering cell therapies more accessible to patients. And in order to do this, the first question is, what is the current state of engineered cell therapies? Most of you know that if we consider, for instance, autologous CAR T cell therapies, these are ex vivo cell therapies, and we have 4 big problems with them, even though they have huge potential to cure terminal cancers. The first problem is technical complexity. It requires substantial patient participation, which is very difficult for sick patients and their caregivers. The second problem is time. We know that there are several reports showing that a substantial portion of patients die or become ineligible for these treatments while they're waiting for their treatments. The third big problem is cost. And here we know that cost of goods per dose is very high as compared to other drugs. And it is, by different reports, around $100,000 for one dose. And access is also a problem. If we map all the hospitals and all the facilities that are that have expertise and capabilities to deliver these therapies, we will find out that there are only less than 2% of all hospitals in the US that can do that. And this means that so many patients have to travel so far to even attempt this treatment. EMBODY wants to solve all these ex vivo problems with one in vivo solution. And the dream is to have a same-day treatment at any US hospital with acceptable cost of goods so that we're able to serve large population, not on the level of ultra-orphan disease. And so in order to achieve this, we scoped EMBODY, and at the high level, we are asking proposers to propose to target any immune cell. We are limiting it only to immune cells. And within immune cells, any immune cell is okay to propose, but we prefer mature lymphoid cells because they're harder to target. In terms of indications, again, any indication can be proposed, but at least one non-oncology indication has to be proposed as well. And this has to do with the idea that the risk that is tolerated in non-oncology indications is much lower. There is low. The safety profile has to be much more robust for non-oncology indications, and that's very important for us. And then the program itself does not focus only on technologies, on designing, for instance, delivery vehicles or genetic material to modify cells in vivo. The program also must address production problems. Reduction of cost of goods is part of that problem, and validation. So having better preclinical models or validation models that are more suited for these types of agents. And this is in scope for EMBODY. Next slide. Okay, so we break, the program is broken down into 2 technical areas. Technical area 1 is platforms and approaches for in vivo or in situ generation of genetically modified immune cells. And this technical area has 2 focus objectives. One is focusing on creating cell-specific delivery in vivo. And the second one is having a genetic material that is not only efficacious for selected indications, but also programmable and controllable. And again, this is because we want high level of safety and ability to control that genetic material in vivo. The second technical area is advanced production and validation of products that are developed using technical area 1 platforms. And here we have 2 focus objectives. First one is about production, and here the focus is on low cost of goods and robust quality control. And the second main objective is creating preclinical validation tools that are more predictive, faster, and cheaper than what we have today. It is important to remember that performers must pursue both technical areas and all objectives. So when you think about your teams, you must have expertise across all these objectives and technical areas. Next slide, please. Okay, so EMBODY is split over 4 phases and 6 years. Phase 1 has to do with design and development of platforms and approaches for in vivo cell-specific targeting, and also developing methods for production and preclinical models or validation models. It is going to be for 2 years, and at the end of this first phase, a program manager will decide which projects to put, which projects will continue to phase 2. Phase 2 has to do with preclinical development and specifically testing selected candidates in preclinical models of disease. And then phase 3 is going to be for 2 years and being focused on one selected candidate and IND enabling study for it. And then phase 4 is going to be focused on first-in-human trial. The scope of the first-in-human trial is quite specific and narrow, so I'd recommend that you read more thoroughly on what is in scope for the clinical trial. It's a limited scope, only for 1 year, and a limited number of patients that ARPA-H is going to fund for successful performers. Then during different parts of the program, for example, after phase 1, we expect that there will be material transfer, know-how, and data transfer between sub-teams of each project. For example, in preclinical model development, some successful models should be transferred to a team that will be testing selected candidates in preclinical model of disease. Or if the model has to do with CMC, then transfer to CMC sub-team. So there are several points at which material, know-how, and data transfer is expected to happen. And finally, we expect that the teams will interact with the FDA, and the expectation is that the Interact data package is ready by the end of phase 1. The pre-IND package is ready and submitted by the end of phase 2, and then the IND application for a successful candidate is ready and submitted by the end of phase 3. And again, here to remind you that all performers must pursue both technical areas and all objectives. So please make sure that you will find and assemble teams that can address all of these. The most critical part of our programs are metrics. And this is because, like Amy said, our projects are milestone driven, they're time bound, and they have specific quantitative and qualitative metrics. And you can find metrics in ISO section 1.3, and I recommend that you read them thoroughly, and when you write your solution summaries, reflect on those metrics and how you can fulfill them. First, we can look at them at the high level. They're broken into categories. Technical Area 1 metrics categories include efficacy, durability, safety, regulatory metrics, clinical, and target product profile. And then technical Area 2 metrics include metrics around CMC, preclinical models, and regulatory. And there could be a situation where you think that the metrics are not meaningful for your particular case, maybe for your selected indication or your technology. In that case, you are welcome to propose your own metrics and describe quantitative improvements that those metrics represent over the state of art, and you must provide rationale. However, ARPA-H does not guarantee that it will accept that rationale. Next slide, please. Okay, so let's look at specific metrics. This is a snapshot from ISO section 1.3.2 on metrics for technical area 1. And this is efficacy metrics for phase 1 of the project. So what we have here is, and I'm going to read it and explain the metrics to you. So the first one is that performers must propose minimal % of targeted cells based on cell of choice, indication, and therapeutic effect sought. At least 2 different indications or disease types must be proposed, of which at least 1 must be non-oncology indication. And that is what I mentioned before. Please make sure that you have at least 1 non-oncology indication. You can propose all your indications non-oncology, could be oncology and non-oncology, could be just 2 indications, could be more than 2, but at least 1 should be non-oncology. Second bullet point says preference will be given to targeting non-phagocytic cells, as it is much more technically challenging to target non-phagocytic cells in vivo. And this is, in this community, you guys probably know that this is, whenever the data are presented, we always see how much more phagocytic cells are able to be targeted. So next, the third bullet point says that for non-phagocytic cells, more than 5% of cells must be targeted in vivo, or the minimum required to achieve a therapeutic effect. And there is an asterisk here because it clarifies what we mean by 5% of cells. And if you read in that ISO, you will see that more than 5%, for instance, of T cells means that of all circulating T cells in vivo, more than 5% have been successfully transduced, and the delivered genetic material can be detected by PCR. So we clarify what it means that we are trying to quantify here. And then the fourth bullet talks specifically about phagocytic cells in the same narrative. And then the fifth bullet, for instance, here talks about reproducibility. We care a lot about reproducibility because we want these programs to be accelerated and become products as soon as possible. Therefore, here we have a requirement for reproducibility. The % of cell of choice targeted in vivo must be reproduced in at least 2 different labs in hands of different investigators, and we prefer an independent lab for an independent validation and verification studies. And you can find those labs, they're available, and make sure you think through this reproducibility requirement as you write your solution summaries. In vitro viability of transduced target cells must be demonstrated at more than 80%. And this is another quantitative metric that we would require. And this is just a snapshot example of metrics, and the table is large. So I would encourage you to read it thoroughly and reflect on how these metrics align with your technology and with your bold vision. Next slide, please. So here is an example for metrics for technical area 2. It has slightly different flavor, but also a table with a lot of details. This is ISO section 1.3.3. So first, technical area 2, to remind you, it combines production and validation. Here you see CMC metrics, and for phase 1 specifically in this snapshot. So you can see that we care a lot about cost of goods. And in this particular case, you can see metrics for cost of goods. In the double asterisk below, you can see what we mean by cost of goods. And particularly here, if let's take an example of a synthetic delivery vehicle. And then in this case, the requirement is that cost of goods per patient should be less than $1,000. What does it mean per patient? It means that if you need just one dose to treat a patient, your cost of goods can be $1,000 or less. If you need 2 doses to treat a patient, then a cost of goods per dose should be $500 or less. And if you need 10 doses to treat one patient, then the cost of goods per dose should be $100 or less. So the idea is that to have a high bar for cost of goods to treat a patient, and then it depends on your indication and your route of administration and frequency of administration to achieve efficacy. The second set of metrics is around validation models. So here in the second row, you see preclinical models. These are general requirements for metrics. We understand that development of models can greatly depend on what you're going to propose. And in the ISO general description, you will see all sorts of examples of what preclinical models could be and what they shouldn't be, and it's a wide variety. So here we have general requirements for preclinical models. For example, you must propose to develop at least one new or significantly enhanced model, at least one existing, and it should be a physical model, in vitro or in vivo, to address 2 objectives. Objective 1 is for this model to be more predictive of responses to technical area 1 agents. And objective 2 is to allow for faster measurements and or being cheaper to implement. Because it's hard to predict what models you guys will be proposing, there are examples for metrics for different models. For example, in row 3, you can see examples for in vitro models. And examples include it has to provide more than 4 a magnitude increase in sensitivity of off-target effect detection, for example, or it should provide more than magnitude increase in number of candidates that could be screened, or in a number of immune cells that could be tested, or in a number of tumor types that could be tested simultaneously. So you can see that the requirement for an in vitro model development is give us a magnitude improvement in a critical parameter that is aligned with predictability, speed, and low cost. Then there are examples in this 4th row. There are examples for in vivo models. You're free to propose in vivo model development. And here are examples. If you're developing a new model, animal model, you may want to recapitulate all mature myeloid and lymphoid human immune cells in the same model and maintain that phenotype for more than 6 weeks. So this is a good example to adhere to for a metric. And so I recommend that you read the metrics tables thoroughly as you think through your technologies and your proposal. Next slide, please. Okay, so there are other requirements that I would like to highlight. There are many requirements, but some I think are worth highlighting here just to emphasize that they're important. This first slide shows non-technical area related requirements, for example, team structure and other. First, every team must have one commercial and or for-profit organization. And any organization may propose to be the prime awardee, but we prefer that a commercial entity is a prime. Other entities, such as academic institutions, may participate as program awardees, sub-awardees, but federal entities may not propose to this ISO as primes or as sub-awardees. Federal entities, however, could be independent labs for independent validation and verification. Specific content, communication, and networking and team formation are the sole responsibility of the proposers. And here, this is one of the reasons why we have teaming opportunity on our portal, because we want to make sure that we understand that we are asking for quite large scope. So we would like for you to have an opportunity to find team members. Proposers may submit only one proposal as the prime proposer, and the group may participate in 2 proposals, but cannot be the prime proposer on both. And this has to do with the fact that we want this is a very heavy effort, and we want to make sure you're focused on delivering on these metrics and on this proposal. If a proposal has one principal investigator, that principal investigator has to commit at least 50% of effort. And if you have co-PIs, then each of them must have at least 20% of effort each. And if you have co-Is, then co-Is must have at least 5% effort each. Again, this has to do with the fact that this requires so much effort to pursue that we need your focus on the program. When we say principal investigators, this is a general term. It could be a professor at the university, it could be a chief scientific officer at a commercial company, or any other VP of R&D, whoever that technical person is who is going to be leading this effort, or a team of technical people who are going to be leading that effort. These are principal investigators for this purpose. A full-time and experienced project manager must be budgeted for in the proposal and must be hired by performers upon successful award to ensure efficient communication between performer teams and subcontractors. So this is very important. I think it's quite natural for companies to have someone having a project manager role, and this is quite unusual for academic institutions in my experience. So we are highlighting this because this effort is so large that requires so much oversight of different activities going on in parallel and getting teams together, that it's critical to have a project manager that is budgeted for. And this person, I would like to highlight, should be experienced project manager. It cannot be a postdoc in your lab, because this role is heavily administrative and organizational, and not a scientific in nature. Next slide, please. Okay, so now this slide highlights some requirements for technical area 1 that I wanted to highlight. So first, it is expected that proposals will require diverse teams with the expertise needed to achieve the goals and objectives. And we expect that TA1 performers will have biomedical engineers, genetic engineers, chemists, and other relevant experts. So think about the diversity of expertise that you need on your team. Performers who are pursuing gene editing must consult with a NIST Gene Editing Consortium representative. And the presentation after mine is from Samantha from NIST, and she's leading this initiative and would be very useful for anybody who is considering gene editing, delivery of gene editing machineries. And this consultations, we expect that will happen at least annually with NIST, and discuss latest advances on methods, protocols, and reporting norms and standards. And I personally think it would be hugely beneficial for our performers to have discussions with NIST. It's very, very useful. Clinicians must be consulted with respect to dose ranges, routes of administration, interim readouts for the first-in-human clinical trial, and other disease-relevant parameters throughout the entire project length. So this is very critical because we want to make sure that you are adhering to the time that is very limited to conduct the research. And it's very important that the vision for the product is being contemplated early on with actual clinicians who are potentially going to be leading a clinical trial. And we know that it can make or break a program if these things are not thought through early in advance. It is critical that an independent team executes phase 2 and phase 3 animal studies to validate technical area 1 agents. When I say phase 2 and phase 3 in this bullet, it means phase 2 of the program and phase 3 of the program, not the clinical trials. So the idea here is that if you develop and you design and develop your delivery agents, for example, you are biased towards naturally towards conducting experiments in animals. And so the idea is that a different team or a different group of people conducts experiments in animals with the agents that are designed by engineers and chemists and other team members. High-level target product profile drafts for selected agents and candidates should be prepared. For commercial companies, I'm sure that target product profiles are absolutely day-to-day activity, and it's very clear what to do. For some academic institutions, for some academic professors, this may not be something that they do. And so this is just a small guidance on what they include. They include considerations for product attributes, indication, dosing regimen, efficacy, safety, and durability targets. So the next one, performers must communicate with the FDA throughout the program. And as I mentioned on the timeline slide, we expect that you prepare for the Interact meeting, for the pre-IND meeting, and then the IND submission at the end of phase 3. And then for phase 4, this is a clinical trial. I mentioned that we have a very limited scope for it. Sorry, not very limited, just limited scope for it. We are requiring that the ARPA-H will fund a clinical trial for up to 10 subjects and for 12 months of the study. I understand that most studies need to be longer, and you may want to recruit a larger group of patients. It may be an extension cohort, that not just dose escalation cohorts, and you may also want to be required to do long-term follow-up. So we just want to make sure that you know that we are funding just this scope, and you probably want to start thinking about complementary support and funding if you want to pursue a much larger clinical trial. Next slide, please. Okay, and so this is the slide related to technical area 2 requirements. Again, these are not all requirements, but something we wanted to highlight for you. It is expected that the proposals will require diverse teams again. And so here for technical area 2, we want you to consider different CMC experts and also model development experts on the team. Cost of goods, as I mentioned before, is a very critical metric, and proposers should articulate how they're going to reduce cost of goods. So be sure that as you're writing this, you communicate with the CMC experts and your team members. You know exactly where those reductions will come from. You think strategically about, essentially from day one, you have to think about your cost of goods. It is up to performers to propose one or more preclinical models to develop. Choose any number you want to propose. It doesn't matter to us, but they have to be physical, so in vitro or in vivo model. They could be systems that can be used for screening, validating, and quality control of technical area 1 agents. There are some examples in the ISO that we are listing in terms of what they are, and you can read them in the ISO. The next thing we want to highlight is that if proposed, performers must demonstrate that they are able to secure necessary infrastructure for animal development and a clear plan of disseminating successful animal models to broader research communities. We are not restricting you what kind of animal models you would like to pursue, just as long as you have a plan for dissemination, you have infrastructure, you have experts who can do that. That works for us. And then please read specific requirements of what not in scope for technical area 2 model development. For example, preclinical models that are suitable only for specific disease indication, they are not in scope. Models that are purely computational or AI ML based and that are not physical cannot be proposed. So if you are proposing AI ML algorithms, they have to be tied to a development of a physical model in vitro or in vivo. And disease models that do not incorporate immune system components are not included in the scope, and model development in non-human primates is not included in the scope. Next slide, please. Okay, so I have 2 more slides. This one is a slide that we put together based on the questions that already came in. And please keep asking questions till the end of day April 22nd. So we may have rephrased some of the questions to maintain confidentiality. So the first question is, can ex vivo cell modifications be proposed? And the answer is no, only in vivo in situ cell modifications can be proposed. One of the reasons that EMBODY focuses on in vivo is because we aspire to eliminate all manufacturing infrastructure and expertise that is currently required for ex vivo genetic modifications of autologous cell therapies. And that aspiration means that no, you cannot propose ex vivo cell modifications. The second question is, can consultants to ARPA-H apply as part of proposer's team? And the answer is no. Anybody who provides any service to ARPA-H cannot be part of proposer team. And I recommend that please make sure that no one on your team has such conflicts of interest. And this is also referenced in ISO 4 section 4.2.3.1. So please make sure, ask your team members, do they have consulting agreements with anybody? Do they provide services to ARPA-H? Be sure you understand what kind of conflict of interest may arise in the future. The third question, who can be prime or a lead? And I mentioned this earlier. Any organization can be a prime, but we prefer that a commercial or for-profit organization is a prime. Although universities can be a prime, we just give preference to for-profit organizations to be primes. And this is also in the ISO section 1.4.1 and section 2. Fourth question, is it required that a prime has the largest percent of effort or largest percent of the budget? And the answer is no. Effort and budget allocation should be appropriate for the proposed projects and tasks and should not depend on which organization is the prime or a sub-awardee. Next question, what is the program budget? We do not discuss program budgets or allocations for technical areas. Please create your budget according to what you're proposing. Should all technical areas and objectives be covered? And I mentioned it 2 times. Yes, absolutely. All teams are required to cover all technical areas and metrics in their proposals. Teams that lack any technical area or sub-area within the proposal will be deemed non-conforming. And the last question that we received, can a small business be the prime for a large team? And will we have to perform the majority of the work as stipulated under the SBIR or STTR? And the answer here is no. There are no requirements for primes or sub-awardees to self-perform any specific portion of the proposed work under the EMBODY ISO. So this is a difference as compared to SBIR or STTR requirements. Again, we will continue to review your questions and we'll post answers after April 22nd. Next slide, please. And the final slide here for me is to highlight the key deadlines that you should be aware of. Again, April 22nd, last day to submit questions through the EMBODY website, or you can send questions to embody@arpah.gov email. May 2nd is the deadline for solution summaries. Between May 15 and May 21st, the decisions on solution summaries will be made and selected proposers will be invited to write full proposals. And then June 11th is the deadline for full proposal submissions. Somewhere around August, decisions on the selectable proposals will be announced. And that concludes my part of the presentation. I think next goes Samantha from NIST, and that's Michael, maybe you can take over here.
Hi, good afternoon, everybody. And I see Samantha's online, so I will turn it over to her. Thank you for joining and thank you for the presentation. Over to you, Samantha.
Hi, everyone. I'm going to try to give you a sense of some things that you should be thinking about, considerations as you begin to develop, and with this sort of program where you're looking towards translation and getting things over the finish line. Next slide, please. I'll tell you a little bit more about NIST in a few slides, but you'll have access to these, and so don't feel overwhelmed, as I'm just going to sort of give you an overview of some of these concepts. So as you all are inventing and coming up with innovative strategies that'll hopefully help so many people, think about the ways in which discovery, translation, scale up. We want these things not to fail and fall into these classical valleys of death that we think about between discovery, translation, or even getting into clinical implementation. Next slide. So one of the things that, as Daria was saying a lot, is about reproducibility and having confidence. And so this is a very important concept that I want you all to think about. And with irreproducibility, there's a lot of resources. ARPA-H is going to be putting a lot of resources into all of your innovative ideas as those that get funded. And how do you think about making sure that what you're doing is really robust and really able to be reproduced as you need it to be in order to have a solid product? So next slide. Okay, so here's one of the little things that I want you to think about reproducibility, but I also want you to think about an expanded concept, which is high confidence. And sometimes reproducibility is not the same as high confidence. So just as you see over here with these bullseyes, you can have precision, which is the first bullseye. Everything's clustered together. And so that would give you the concept of reproducibility, but you don't have accuracy. You want those to be over a value that's what you mean to hit. And so when you think about this, I want you to think a little bit more broadly with a little bit more complexity about things where, how do you know if the answer's right? And not just if you can reproduce something, because you might actually have a systematic bias. And if you can account for that, you can scale. So how do you know whether you're getting true biological signal? There are a lot of things in a process. These are very complicated things that you're trying to do. And you want to be able to say that you're getting biological signal and not just all the very many ways that you are getting noise in your system. Next slide. So whether it's the EMBODY program or other programs, whatever science you're doing, here are some questions that I want you to think about. As you're collecting data, as you're developing, as you're translating, how confident are you that the answer is correct? What's the consequence if it's wrong, and what can be done to understand if the answer is correct? Next slide. So where can variability happen in your process? I want you to think of your entire process as you're envisioning things, as samples are being shipped to you, your key components, as you're storing them, as you're doing everything in the lab, in your initial buildup, as you're thinking about translating that, how are you going to deploy that in vivo, anywhere along your process. Next can be places where you have variability. Next, so how are you going to de-risk this? Next. So all the way across this process, you can have tools and strategies that will help you. And I'll just give you a little bit more information about this, but validated methods and protocols, some things, some sort of ground truth, if you have that, that's available, that can be very hard and unavailable. So we'll talk about that a little bit more. How do you know whether your instrument's working? What are you using to make sure that that box, that sometimes we trust a box, is actually within its performance range? So you want to have some strategies there. Your biological materials, you want to have some specifications as to what those should be, and analysis. How are you making sure that all of that information is being combined in a way that you can responsibly use it to make the key decisions that you need to make? Next slide. Okay, so NIST talks about this word measurement assurance. You don't have to use that word, but basically, in a nutshell, it's how do you know whether you can use the information that you have in a confident way? Are you confident that you can make decisions and important decisions based on your data? Next slide. All right, so there are lots of strategies for this, and this isn't to say that you have to use all of these strategies or there's any right answer as to a combination of these strategies, but these are just approaches that you can use and pick the ones that are most meaningful for your approach to get your confidence. So, where are you having the possibilities that things might impact what you're doing? Have you thought through your design of experiments, orthogonal techniques and measurements, control materials or reference materials that have some ground truth that you can use to know whether you're getting the right answer, cross-comparison studies, that's going to be a part of the requirement for the EMBODY program, where you need to be able to translate across and have another lab perform that same process. In-process controls, which are a little bit different than reference materials, performance specifications, all of these things in combination and different combinations can help you have more and more and more confidence that you can trust your answer and that you can be assured that you're making decisions based on content that you feel you can trust. And so here's some things on the side you think about. If there was a problem, is your assay or approach sensitive to detect it? How do you deal with that if you do find an issue? And you always have to think about this. It's for your fit for purpose. Sometimes people will say, it would be really great if you just said do 1, 2, and 3. And sometimes that's challenging because you don't want to be limited. You want to pick the things that make the most sense for your use case, the technology that you're developing and the way that you're deploying that. Next slide. Okay, so this is one of those things. This is the kind of thought process that isn't the most fun. It's not the most exciting, but these are the kinds of things that if you don't think about early might predispose you to be sliding into that valley if you don't de-risk some of these things very early on. So this concept of knowing thy assay, what are you measuring? And this might sound like a very obvious or trivial thing to say, but are you sure that your assay is able to measure the feature that you want to measure? So if you want to measure fluorescence, you probably don't want to use, an agarose gel and look at DNA size. You want to use something that's going to give you, you're probably going to need lasers if you want to look at fluorescence. So is your assay really measuring the thing that you want to measure? What are key variables, things to de-risk, including personnel? Can't stress this enough. Usually people are one of the biggest variables in your process. And so how do you make sure that they have the right information, the right protocol, sufficiently detailed enough protocol in order to do the things that you want to have done in a reproducible and robust and trustworthy way? Reagents, can other labs get the same results? And most importantly, even if you can measure something, is the output biologically meaningful? Just because you can measure something doesn't mean that it's giving you information that's actionable. All right, next slide. Okay, sorry about this. Sometimes the slide goes a little bit awry. The very first box is assay qualification. The second box is assay validation, and the third box is reference materials. So ground truth is really hard to find in biology. So what you can do to understand whether your measurements are right, whether the process is giving you good information, is first you want to qualify your assay, your measurements. If you're trying to develop a product here, you're trying to develop therapy, what are the ways that you're using to characterize that therapy, characterize whether it's working? And do you understand the limits of those assays? If you need to get a value that is meaningful for you as a value at 10, if your assay can only give you information from 20 to 100, then it can't do what you need it to do. So knowing whether your assay can really do what you need it to do and what's its performance range. So that's when we talk about qualification. If you're talking about the word validation, the way that you use this word is that you're setting criteria, saying, I now know that my assay can only do from 20 to 100. But I need to make sure that I can constantly hit 50. 50 is the requirement. 50 is what I need to do. That's the acceptance criteria. So if I'm below 50, if the signal's 30, it's not good enough. So you're setting specific criteria that you're validating that your assay is doing this exact thing. So that's validation, which goes on top of qualification. What is the possible range? And then what do I absolutely need it to do and hit each time? And that's where additional controls or additional reference materials, something that helps you know, are you getting that right answer? And so the last box is supposed to be reference materials. I'll try to fix that for you all. So this is that ground truth. You don't always have it. Reference materials can be physical things, they can be data, but what do you need to help you know that you've actually got something to compare against, that you're getting the right answer? Next slide. Okay, so I'm not going to go through this in detail, but is the data good quality? This table is just something that you can think about, splitting this concept. A lot of times everything gets wrapped up in this word reproducibility. And so I want you to think a little bit more deeply about this and say, well, reproducibility is just, am I getting the same thing, replicates, maybe operator to operator, day to day? It's not the same as accuracy. It's not the same as robustness, where you're saying, if I have a temperature change in that room, if it's a different donor, if it's a different physical location, hospital, how robust is my process to things that might reasonably change or be different when you're trying to redeploy something. And how do you de-risk that if it's not robust to it? Know the kinds of things that might really happen and try to test those and know whether your system can tolerate those changes, because they're real things that you might have to deal with. Limits in detection and reporting, I'll just stress that one of the biggest things is not having enough information or details. Reporting, documenting a full process is one of the most enabling things that you can do. Next slide. Okay, so here's an example of an assay qualification process, just for you to think about. Do you have the availability of any control reference material, reference data that allow you to understand something? Where are places that this could go wrong, or variables that you need to de-risk, whether it's a new vendor, whether it's shipping, whether it's a new instrument, whether it's a new operator? What do you need to de-risk? Refine protocols and approaches to minimize that risk, give you more confidence. Get to baseline data now. Now you've done a lot. What's baseline? How do you know whether something's deviating from what you expect to happen? And can you add any additional controls or approaches to give you the best confidence possible? Next slide. So here's just one example of reference materials, especially with this kind of program. I imagine there's going to be a lot of sequence analysis, a lot of characterization. So I just want to put this out there as one particular control material that is available through NIST and work that NIST has done that are available to you. These are whole human genome sequenced genomes that have additional characterization, phasing, large variants. This is work that was done between NIST initially and the FDA, and then a larger consortium working with all sorts of people in the community, especially those that make DNA sequencing technology, so that you could have highly characterized full human genomes in bottles that you could use to test and make sure that your DNA detection technologies are working. So this is actual ground truth genomic information. And so here's just an example of how you can couple a reference material into a process, have some truth, and know whether something is deviating as you're trying to use a key technology and make very important decisions based on that data. Next slide. Okay. So what if you don't have a reference material available? What are you going to do? And this is where, as you all are innovating and maybe inventing things, and there is no such reference material for that, you come up with your own strategies, your own materials in-house developed that you can use to say, here is my something, and I'm going to use it each time. I'm going to put it on each run, and I'm going to create my own expectation that I have something that's constant, that's allowing me to know whether something's deviating in my process, because there's no generalized material. And as you develop something that might be fit for your particular therapeutic application, we will always need to have something that's very close to, if not the same thing as your particular material, even if you can take advantage of community-based reference materials. So continue to think about that. And please, 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 if you do develop something like this, please don't use it all up before you qualify a new batch of it, even if it's your in-house material, because you will need that material to make sure you can qualify a new batch as you use your own in-house material and your own in-house strategies. So it's not that you have to have a reference material, but you should have strategies that are meaningful for what you're trying to measure for your product, that allow you to know whether things are deviating. And as much as possible, where community reference materials are available, I would encourage you to use those. Next slide. Ground truth, there may be ground truth, there or may not, but have something that you have control over. And then there's experimental design. So one of the things here is that No volume of data, replicates, or statistics can make up for a poorly designed study. So in this little cartoon, you've got a fish, they're whacked with a magnet, and they're not very happy anymore, if even viable anymore. And the conclusion is interesting. There seems to be significant decrease in heart rate. The fish must sense the magnetic field, right? And as you can see, this wasn't designed to tell you anything about magnetic fields. So even if you have the right components, if the experiment's not designed to give you the output that you're trying to measure, there's nothing that you can do. No massaging of the data or stats is going to really give you that information. So you've got to really think about, can this give me the piece of information that's going to be meaningful, actionable to me as you go to deploy these things? And is it telling me about as we said earlier, is it telling me about the biological thing, or is it telling me about some other process or some other noise that I might be detecting? Next slide. Okay, so you can have identifying sources of variability. You can do this if you have a problem or something that you want to avoid. These cause and effect diagrams can be a way of just mapping out what are the kinds of things that you need to de-risk, where can you have variability, and allow you to think about what are you going to do to try to avoid that or de-risk that in as much as you can. Next. So this will just be one that you can take a look at as an example of a completed kind of cause and effect diagram. Next slide. Okay, sorry about this too, in the porting over. So the top is all you really need to know for this is that NIST, now I'm going to transition to giving you a little bit about NIST, how we might be able to support in this space, and some of the stuff that we're doing that might be relevant to the things that you're thinking about as you're contemplating proposing for this program. So NIST, we're the U.S. National Institute of Measurements and Technology. So a few things about NIST. NIST is non-regulatory. We don't primarily function as a funding organization. And we are therefore able to work hand in hand. We're the measurement people for other federal agencies as well as for the US. So that allows us to work with academia, it allows us to work with industry, as well as other government to help with measurements needs. And NIST is also part of the US Department of Commerce. So the idea that we're supposed to be helping to support and underpin the US economy. Next slide. Okay, so when you think about standards, NIST, National Institute of Standards, standards is not a dirty word, it's not a scary word, it should be enabling, not inhibiting. So standards help you to know whether you can trust your data. You don't want to make important decisions based on data that's unreliable, that maybe doesn't mean what you think it means. And that's how you should think about standards. How do you have all of the infrastructure to say, do I trust this? If the data isn't good, it's a very good thing to know because you don't make important decisions based on it. So it could be physical things, it could be common practices, it could be requirements, anything that helps you, you can think about potentially becoming a standard or norm that helps with your process accelerate. Standards should help accelerate development, not hold it back. Next slide. Okay. So as you're thinking about things and the way that we're thinking about things at NIST, we're trying to know where in people's processes is it really important to know that the answer is right and reliable. Where in the process is it currently difficult to know if that's right or the data is reliable, and what can be done to give more confidence? That's the kinds of stuff that NIST is trying to figure out, and defining this problem for different areas in the biological science spaces is where my area is. Next slide. Okay, so how does NIST work with communities to meet needs? So there's initial research, translating to industry. In this case, there may be a lot of industry that's going to be involved from the beginning of this, but you're still developing, translating, getting to somewhere like public health and safety. And as I just mentioned, NIST can work and does work with anybody, both nationally and internationally, as there are other standards organizations across the globe that we work with and coordinate with. Next slide. Okay, and because this is something that I apologize again, these shifted a little bit. So how does NIST collaborate on standards? Well, one of the things I wanted to point out, since this is a requirement for this program, is that, again, NIST is also the measurement people for other government, and we work directly with FDA. And in particular, to this conversation, we built this slide with FDA CBER to describe how NIST and FDA collaborate together on measurements where FDA doesn't have the same flexibility to work with people and talk with people the way that we do. So NIST can engage, but then work with FDA in this collaborative cycle to help with the measurements controls that enable products to move forward and the regulators to have a better sense on if they can trust all the information that's coming to them. Next slide. Okay, so if you're thinking about NIST, here's what I would love you to think about. There are many things going on at NIST. I'm just going to give you a quick taste of some of the kinds of competencies, the kinds of areas that we're working in that you may think, hey, are there things that would help me have confidence in some of these technologies? So we're working on measurement assurance across various things in biomeasurements, as you see on this slide, including, as I'll tell you a bit more about genome editing measurements, but cell counting, viral vector work, viability, the kinds of things that you're thinking about where your cell is your product in this program, even though it's in vivo, these kinds of things that how do you understand this since you are most likely starting with some of these things ex vivo. Next slide. Okay, again, just so you have this as a reference, some of the general areas where we're applying some of our capabilities in case any of these things are things that you're thinking about or want to deploy, that we might have some controls, some approaches, some information that could help in these areas. Next slide. And as we come back to reference materials and control materials, we may have NIST reference or control materials that can help you. We have things that are more standard in terms of extracted things in vials. And then we're also thinking towards and trying to develop this new, 21st century control materials. We talk about them as where the materials might be whole cells or living materials. Next slide. And just so you know, in case you're interested, we have some NIST consortia, Genome in a Bottle, it's still active, the Genome Editing Consortium that I lead, flow cytometry standards that now has a new working group on gene delivery systems, and a rapid microbial testing methods consortium that are all still accepting membership in case you're interested in any of these things that I can help point you in the right direction. Next slide. Okay, so evaluating performance, I'm just going to give you a few quick examples of some things so you have a sense of how this might actually come into your hands, what you might actually interact with. So here's an example of how do you know if cells are your dose, or you're trying to figure out how much of a dose to give, and you're starting with things in the lab. How do you think about this? And this was primarily for ex vivo kinds of therapies that NIST has. There's now an ISO standard for cell counting next. And so an approach as well as things that you can deploy. It's actually not a physical material, but it's actually a statistical approach in how you handle your samples and prepare them and then deploy them for measurement that allows you to have clear understanding of the proportionality for your cell count. And so this is now an international standard for cell counting, including the analysis tool that was led by NIST. Next slide. Next, great. Design of experiments. So here's an example of, this was a real study trying to do a cross comparison where there was on the left a lot of difference. And then NIST came in with some design of experiments, testing some of these assumptions. Some of this was actually where things were on a plate. Some of this was actually BSA and everybody needing to source locally because of limitations. Some of this was international data, and then realizing that that reagent was performing very differently in this assay. And it wasn't nominally the same reagent that people were using, and figuring out a way around that, testing that, and then on the right side, really being able to see that data coming in line, and then being able to tease out even more some of these things that were going on based on how that test was designed to identify where the variables were coming from. Next slide. Okay, and then maybe just 1 or 2 more minutes, I will tell you about the work that I lead in the NIST genome editing program, where we are working on physical measurements, data and metadata documentary in order to help increase the confidence of utilizing genome editing technologies in research and commercial products. And what's driving most of this use case is the commercial product of human therapeutics that are using genome editing technologies as a mechanism. Next slide. So if you're genome editing overview. Maybe you've thought about this a lot. Maybe you're thinking about using this technique as you're proposing. There are many different genome editing molecules, and I'll extend this. We can have editing, permanent changes now. There's been so much advancement and deployment now of epigenetic engineering as approaches, and that might be something that you're considering, but you're taking an approach. You've got your cells of interest in this case. You certainly will get to in vivo. You're hoping that you're getting a change that's meaningful for you. You've got to deliver. That delivery, it's small, but it's not insignificant. It's a very big piece of this. So next slide. So if you go ahead and click next, so where's this coming in? There's innovation. Next, we're coming in with the measurements and controls. Next, the data and control data sets. Next, and metadata. These are the kinds of things that NIST is trying to help in this particular area. Next slide. Okay, so where's genome editing happening? So here's some of the stuff. If you're going to use this approach, where's genome editing happening? Is it happening on target? Next. Or is it happening somewhere else in the genome? So you need to be able to report where it's happening. Next. And then you need to know what edits you're making, whether these are DNA edits or if they're epigenetic and you are influencing expression other places in the genome. What are the changes that you're making on target and what are the changes that you're making off target? And if you are going to use an editing technology that changes DNA sequence, FDA requires that you report on target what edits are made and what relative frequency, off target what edits are made, where the edits happened, what edits, what relative frequency. And then for in vivo, as you'll get to in this program, there are other additional considerations on tissue, off tissue, amount of activity. So those are the kinds of things, if you're into this space, that you should think about. Next. All right, so genome editing outcomes can be complex. So again, even though this looks like a Petri dish, you can envision this as this could happen in a Petri dish. This could be what's happening in vivo. And if we're just considering only one set of cells, even though there are many cell types and many tissue types, if we look as small as possible, this is the kind of thing that can be happening. So you've got your editing molecules, you've got your source cells that you're hoping to edit. You might have edits you want, you might have edits you don't want, you might have no editing. And even if we just think about this as on target, and all of this can happen at any places that you do get off target. Okay, so if you go next slide. So you want things to change in the way that you want, but you can get changes that you don't want. I want to say here, just because you get changes you don't want doesn't mean that it's not fit for purpose. Everything has to be fit for purpose, and that's not something NIST will tell you. That's something that you have to figure out. What is your purpose? What can you tolerate? What do you think is a risk? What is safe? And you'll have to do these measurements, define that, and make your case to the regulators as to whether you feel like this is safe to move forward with. But they're going to require you to provide measurements. And then it really depends on what is the true risk, the biological risk to that being present. So again, less off targets, it's probably better, but it really depends on what exactly the implication is of any particular change in the genome, which may be nothing, even if it is very present. So, you're going to have to make that case and be comfortable that you feel like you can understand whether there's any knowledge to think that a DNA sequence change on target, whether you want that or not, or off target, has any biological implications that would be concerning. Next slide. Okay, this is just to give you a sense if you're thinking about this, and this has been thinking about this for a long time, and places where we have identified needs for controls and standards. While we can't work on all of them at the same time, we are working on some of these things. Next slide. Off-target assays. This is something that is not as much in everybody's hands because these are very complicated assays, and they need a lot of really dedicated attention if you're going to run these assays. And so this is just one example of previously how NIST has collaborated with another kind of ARPA program. In this case, it was DARPA, where they invested in the generation of a new off-target assay where they wanted to get at an assay that could give some account for genomic variation, something that could say, okay, we've got all this variation in this population. Could there be an assay that allows us to account for that in one? And OneSeq was one of those approaches. And they funded this assay. But as it came forward, the question became, is this assay really doing what we need it to do? Does it actually work well? And so this was a place where NIST worked directly, funded by DARPA, to collaborate on evaluating this DARPA invested product, getting deep into this, looking at the protocol, looking at the bioinformatics, running it in NIST's hands, adding additional in-process controls, doing tests for reproducibility, and developing a detailed SOP, and actually being able to work between academia and industry in this case towards translation of this federal government invested funded product, which is what was enabling NIST to work on this, because it was confidence in something that the federal government had invested in, which puts it in the purview of something that NIST can do. Next slide. And just as we talked about, if you're unaware of this, if you're doing genome editing, there's a paper that's come out of NIST, just having people think about, what's your starting point considerations, points of failure to consider. So we did a cause and effect diagram. A simple one, doesn't account for everything, but some things that are kind of general that people might want to think about if they have a failure mechanism of editing. Next slide. And again, this is a first. Do you have any, are you reporting sufficiently or recording sufficiently about some of the things that you're doing? This isn't exhaustive, but this is a first report out from NIST of some key things that you probably want to keep track of if you're doing genome editing at a very base level to think about reproducibility. Can you redeploy that experiment? And some of the gaps that we found when we mined the literature. Next slide. And one thing, I'll just wrap up with this NIST genome editing program that I lead. A big activity in it is a NIST genome editing consortium. There are over 50 active members now collaborating. This is other government, industry, academia, working together to try to develop physical measurements, data and metadata norms and terminology norms to help suit the genome editing community, raise all boats, push people forward. Next slide. So just to give you a sense of some of the things that we're doing, we're making physical control materials within our working group 1. We have 2 new working groups, qualifying off-target assays and quality of genome editing components, which has a primary focus on quality of synthetic RNA. That is one of the key components in a lot of the genome editing, particularly CRISPR-Cas-based genome editing approaches that people are using. Working on data and metadata norms that we are piloting now. And there is an ISO standard now. There is a genome editing vocabulary international standard now that is part of work that came out of this consortium. Next slide. Okay, and just to give you just one little taste of this, here is a very brief, snapshot of some of the work we did, a first interlab study where we made control samples that had key features. We got these into the lab hands of participants. You can see some of these technology makers and users here, which are people that make technologies, people that are trying to make therapeutics right now. They got the same samples, they analyzed it, they gave all this information back to NIST. All of this is going to become publicly available. And this information is supposed to help with data sets and a pilot kind of control approach, if it is useful, that really gets us at everyone has the same sample. How do we understand whether the DNA measurement technology is giving comparable information and limits of detection? Next slide. All right, so here's my last slide. I know I've said a whole lot to you, and it's a lot to digest, but you'll have a time to look over this some more. And you're not really meant to read all of this right now, but I wanted to leave you with, as you all are developing, as you're thinking, as you hopefully make really exciting technologies that are going to be paradigm shifts, what do you think about that it's going to help you get there, right? And this is just a list of some things to think through. Right? What types of assays or technology have you generated? Here you're trying to make a therapy, right? But you might have some iterative kind of measurements that you need. Who's the intended operator of your technology? Is it supposed to be, a mature company? Is it supposed to be the every, lab technician at that hospital? Who is supposed to be the operator? Where are you getting your components? your genome editing molecules, your viral vectors, your LMPs, whatever you're going to use, where are you getting these things? Is there any oversight or authority? How do you know whether you're take your process, break it down into smaller unit operations, and how do you know whether that unit is successful or failing? And fail early, fail fast, and figure out how to fix that, and make sure you don't get to the end of a process before you realize something's wrong, right? Quality of your components. Have you recorded things accurately, sufficiently detailed? Can somebody else deploy this? That's a key part of this program. Think about that early. How are you going to train new staff and operators? How are you going to communicate this so that not just you all that are developing, but the other people that need to do this are going to be able to operate it? And where can you minimize human error? So there's more of that here. But these are just some thoughts. Prime your mind to hopefully have a lot of success in what you're trying to do. So this is my last slide. Actually, there's one more slide, but it's really just a few takeaways. But that's kind of the end of my messages for you.
Appreciate it. Thank you for that presentation. That was excellent. And with that, Georgina, if you wouldn't mind.
Hi, good morning. My name is Georgina Perry. I'm an agreement officer under the Business Innovation Division. The purpose of this portion of the presentation is to provide you with an overview as to why the ISO was selected as the solution method for the EMBODY program, and to hopefully help answer some of the questions you may have as you're preparing to draft and submit a solution summary, and subsequently a full proposal if an invitation is extended. Next slide. Some of the ISO basics. The ISO allows for merit-based awards to be considered. The ISO allows an opportunity for award selection to be merit-based and for technical quality solutions to be weighed more favorably than price, which is one of the reasons why budget range or ceiling was not established. Best ideas are more important than price. The ISO will follow a competitive process where the best ideas will be selected to move forward from the abstract stage to the full proposal process. Each proposal will have a unique statement of work that aligns to the proposed solution. ISOs allow for a variety of approaches to one problem, not one singular solution. The ISO recognizes that multiple solutions are available to one problem, which is one of the reasons why traditional FAR-based procedures will not be followed under this ISO. The proposals will not be compared to one another, and each proposal will be evaluated independently based on their own merit and proposed idea. ISOs are flexible and allow for non-FAR-based instruments to be considered and negotiated, such as other transaction agreements. Next slide. Award types. The award types that are considered under this ISO are only other transaction agreements. We will not be contemplating cooperative agreements, grants, or FAR-based procurements. Awards to commercial organizations are preferred. There will be a maximum of two proposals considered for any one performer. One is a prime and one is a subawardee. Awards will include fixed price based milestones with payments tied to actual milestones accomplished. The award timeline. Full proposal evaluations are expected to be completed by August of 2024. ARPA-H will begin negotiation with those selected to move forward with an award in September of 2024. Those negotiations would include the statement of work and the terms and conditions. Award funding. There is no funding limit, ceiling, or range for individual awards and for the overall ceiling for the EMBODY ISO. All conforming proposals will be evaluated and considered for an award. Traditional awards, once again, will not be considered, so any FAR-based procurements, grants, or cooperative agreements should not be proposed and will not be considered. The overall process for the ISO. The solution summary is identified in page 27 of the ISO. The solution summary submission is mandatory. The length should not exceed 4 pages. This excludes the cover page and the ROM. The ISO solution summary should follow the format outlined in Appendix A and should be submitted to the link provided within the ISO Full proposals are also identified on page 27 of the ISO. Full proposals are by invitation only. So unless you receive an invitation, please do not submit a full proposal unless you receive that invitation. Full proposals are to be submitted through the ARPA-H Solutions website. Summary of the proposal and detailed proposal information should not exceed 30 pages. This excludes the statement of work. The evaluation and selection is identified on page 31 of the ISO. The government will review each conforming solution against the criterion identified within the ISO. Criterion 1 and 2 will be evaluated for each proposal against criterion 1 and 4 in descending order of importance. Selection for award will be made as outlined within the ISO. The only type of award that will be considered, once again, is the other transactions. And what is an other transaction? ARPA-H has authority to award other transactions when the use of such authority is essential to promoting the success of the project. Other transactions are a form of contracts. However, they will not be FAR-based. There will be acceptance, consideration, and legality to these other transaction agreements. OTs are designed to reflect commercial contracting practices rather than traditional federal procurement contracts or financial assistance vehicles. Other transactions are collaborative. There is an expectation that throughout the life of the other transaction agreement, there will be collaboration and partnership with the government and the performer throughout the length of the contract. Free-flowing negotiations are less restrictive than FAR-based procurements and financial instruments, which is one of the reasons why OTs are the preferred award vehicle under this ISO. The evaluation criterion under this ISO. The first one is the overall scientific and technical merit. This is applicable to the solution summary and full proposal. Criterion number 2, which is a proposer's capability or related experience. This is applicable to the solution summary and full proposal. Criterion number 3 is potential contributions and relevance to the ARPA-H mission. This is only applicable to the full proposal. And criterion number 4 is a price proposal, and this is, once again, only applicable to the full proposal. We ask that you monitor SAM.gov. Any changes made to the ISO will be made via formal amendment and posted to SAM.gov. No information discussed at today's proposals day should be construed as modifying the terms of the ISO. We ask that you read the eligibility requirements within the ISO and conform to those requirements. Non-conforming proposals may not be evaluated and considered for award. And dates and deadlines to keep in mind is that the solution summary is due May 2nd by 5 p.m. Eastern Standard Time. Proposals are due June 11th at 5 p.m. Eastern Standard Time. Any questions that you may have regarding this ISO, we ask that you submit them to the EMBODY email provided here. Some of the frequently asked questions that we received with this ISO, we ask that many of those questions could be answered by reviewing the ISO. So prior to submitting a question, we ask that you read the ISO, you read the frequently asked questions page, along with the answers that are being provided via the SAM.gov website. All inquiries should be submitted to ARPA-H, to the ARPA-H Solutions website. We ask that you write those questions as clearly as possible. Do not include any proprietary information within that question. ARPA-H may rephrase some of the questions for administrative purposes only. And if there's any contradiction between the frequently asked questions and the ISO, we ask that you defer to the ISO. Okay, and next I'll go ahead and turn it over to Michael for the final portion of the presentation.
Hi, I will take us home and hopefully end early and give you some time back. So with the review process, as Georgina mentioned, there is a solution summary. That'll be the first part that will be a kind of a gate review, if you will, where we will invite you or not invite you for a full proposal. So put as much detail and content as you can in those 4 pages. And then if you are invited, you'll continue to the proposal stage where you'll have 35 pages to elaborate on your solution summary. And our team will work to make SRO recommendations, and then we will notify you, as we mentioned, roughly in August, and begin negotiations sometime in the fall. In addition to the ISO, there will be what we call our other transaction bundle. So the transaction bundle will include the technical management section, the task description document, cost proposal. We're going to provide you some Excel spreadsheets. We ask that you utilize all these tools and provide us with the information that we're asking for in those. And then there's going to be a number of administrative and national policy requirement documents. As Daria mentioned earlier, conflicts of interest, IP, and obviously in this case, if you're doing human subject research or animal subject research, those documents will need to be included as part of your proposal. Some suggestions and reminders. As I just mentioned, the cost estimate of funds requested, any cost sharing, those are important things. Cost sharing is not required. Costs should be broken down by fiscal year. You'll see this in the spreadsheets that are attached, including backup documentation. For any cost over $5,000, this number may change, it may go up. As well as itemizing subcontracts, making sure all cost proposal documentation is prepared, making sure we have the good level of detail. Make sure that your proposed costs are realistic. Obviously, you don't want to spend millions of dollars on management and a few dollars on science, so focus on that. How are they consistent with your statement of work? And make sure that your technical effort is appropriate and your travel costs are appropriate, etc. We'll provide you with a checklist. Please use it. Make sure that you use the checklist. You don't change the formulas in the spreadsheets that we provide you. Don't delete columns, rows, because you'll break it. And then, most importantly, don't cost strategize. We don't put emphasis on cost, but we want to be cost competitive. And so by strategizing and pulling all the levers that you have at your disposal when you're building a cost proposal, make sure you focus on delivering what the ISO asks for. I'm not going to read this, but this is your proposal checklist. Some things to highlight. Obviously, in the solution summary, you're only going to provide a ROM. In the full proposal, you're going to provide a full cost proposal with your vendor quotes and subquotes. And then if there are any consulting agreements or arrangements, make sure those are as part of your cost proposal as well. And so with that, that brings us to the end of the presentation. I want to thank everyone who presented for their wonderful presentations. And I want to thank you guys for your time, taking the time to listen to us. And we look forward to seeing your solution summaries. Remember, they're due on May 2nd. So thank you very much. With that, we will end this meeting. And so have a great, wonderful day.